Anyhow, this is part 2 of this week's lecture, and hopefully it won't freeze. And I'm going to be talking, I talked about basically the microbiome earlier in the week, about the bacteria, some of the fungi biota, and now I'm going to talk about the viruses. The viruses or virome, our viro microbiome. So anyhow, viruses, virusoids, and prions, which are subviral particles, we know are not cells. They're acellular. Even though they are not cells, they actually would infect all forms of life. So they are viruses that will infect plants, animals, protozoa, even bacteria, and that's one thing that we all know, that they are infect all forms of life. How they do that is basically they will enter the host cell of that living organism, and then they take over the mechanical or the metabolic machinery of that host cell to make more of themselves. They replicate using the host cell's machinery. Now, is this true reproduction, or is it just being replicable because of using the host cell's structures, such as ribosomes or enzymes, to make more of themselves? The bacteria or the virus also has basic structure. Those first 2 bullets show or name the structures found in all viruses. First of all, they've got to have a genome. The genome is either DNA or RNA, not both. You will find one or the other in a virus cell or in the virus. And the genome can either be DNA, it can be single-stranded or double-stranded, or circular, or RNA, which can be single-stranded or double-stranded RNA, which can be linear or circular. So anyhow, that's an important thing to remember about viruses. Their genome is DNA or RNA, not both, like you find in prokaryotic cells or eukaryotic cells. Anyhow, many of these viruses come with a protein sheet that surrounds the genome, known as a capsid. So a capsid will surround the genome. The last 2 features aren't on all viruses. They're found on some. Some viruses have this membrane surrounding the capsid. And this membrane-like feature is called an envelope. The viruses actually pick up this membrane from the host cell which they've infected. It picks it up from the nuclear membrane or the cell membrane after it's been assembled, as it leaves the host cell. Again, and some viruses may have these surface molecules, glycoproteins, that are on the surface, and they're referred to as spikes. The function of these is usually for attachment or for attaching to a host cell. So anyhow, on the screen here, you'll see that we've got on the left, we've got a naked virus. That is the term given to a virus that has the genome and a capsid, okay, the 2 basic structures. On the right of the screen, you're going to see we've got the enveloped virus. So that will include the capsid, nucleic acid, its genome, and then also that membrane-like envelope. That is called an enveloped virus. Okay, now all living things can be infected with viruses, even bacteria. But bacteria, viruses, they have a special name. They're called the bacteriophage. So if you see that word, you know that is a virus that infects bacteria. And that virus is a little more complex in structure. It's got more than just the capsid and genome. It does not have an envelope, but for the head, you've got the capsid, which encloses the viral genome. It has like a long neck. That is the sheath part. And then for the base, it's got tail fibers and a base plate. So these are used for attaching and help the genome enter the bacterial cell. Now the interesting thing is we've got bacteria we know in our microbiota, in our microbiomes. Well, also along with this bacteria, we do carry bacteriophages. We do have viruses, bacterial viruses that are part of our human microbiota. So just because the bacteria are there, they're going to have the viruses too. There are some basic viral shapes. We've got 3 basic capsid shapes, and the first one is helical. So the capsid can be in a helical form, which kind of wraps itself around the genome. And that first picture there is showing you the tobacco mosaic virus. This is a virus that infects the tobacco plant. Another shape, capsid shape, will be the icosahedral, also known as a polyhedral. It has many sides. It's almost geometric. I kind of like to compare it to, facets on a diamond. And, many faces, geometrical looking. And this is found, an example here is on the human rhinovirus that causes colds. That will have your icosahedral or polyhedral capsid. Finally, we've got complex shapes. And these are really the lack of either one of the helical or icosahedral capsids, or they have a combination of those. So your bacteriophage is a complex shape because it does contain a capsid and other sheath and other structures. In this last one, the variola virus, this seems to be lacking either icosahedral or the helical capsid. It is enveloped though. It does have the membrane surrounding it, the envelope surrounding it. So anyhow, those are your 3 basic types of viral shapes. Now another thing that's important is not only the shape, what they're made of, but also their steps, the steps in it making or it reproducing or being reproduced, making more. That's what is important. Viruses, bacteria, anything. Basically, get in, reproduce, make more of yourselves, and go on to infect. In this case, viruses do infect. So anyhow, what are the 5 steps? We've got recognition, entry, biosynthesis, assembling, and release. So with recognition, the virus will recognize a susceptible host cell and attach to it. It recognizes a specific cell by the receptors and by signals that the host cell may be giving off. Once it is able to recognize, it will attach. Then the next step is it attaches, it will have to enter the cell. And many ways in animal cells, it enters by basically being engulfed, or the cell itself engulfing the virus as if to eat it, endocytosis. Once that virus is within the host cell, especially if it's been enveloped, it's in an envelope, it will have to release its genome, into the host cell, either the cytoplasm or nucleus. Depending upon the virus, it may go one or the other place, or even travel between the two. But in this case, once it enters, it will have to release its viral genome, And then once the genome is released, it has to be made. The parts will have to be made. And that is the third step of the virus, quote unquote, life cycle. So we've got recognition, attachment, entry, and now biosynthesis. So that means either new DNA or RNA and proteins will have to be made. This is all done by the host cell structures, their enzymes, their stuff. So once the virus or the viral parts are made, they have to be put together. That's the fourth step, assembled. They do assemble within the host cell. And when they're done, they have to be released. They are released. So anyhow, these are the 5 steps, and it would be important that you realize these steps in the order in which they occur. It may come in a test or a quiz, okay? So anyhow, now this is where it gets really pretty complex. Even though viruses are simple in structure, how are they classified? They replicate or are replicated very, very quickly, and there are many of them, and they also, because of the vast numbers, there is a good chance of mutations. So they are replicated quickly, they mutate quickly, there are many new ones in a short amount of time. So they've got to be classified to keep it kind of simple, just to keep them organized. So there are 2 methods or 2 ways in which they can be classified. This is the Baltimore classification, which takes the approach that viruses are non-living infectious particles. And in this method, they are classified by their type of genome, okay, whether it's DNA or RNA. And also, they take into account the process by which messenger RNA is made, okay? How the messenger RNA is made. And because of these 2 criteria, viruses in this classification system are broken into 7 groups. And the 7 groups are listed here, shown here in the diagram. Group 1, Group 2, Group 3, Group 4, 5, 6, and 7. Now, you'll see in the lighter purple in the box, that is giving their genome. And then the dark purple boxes are examples of viruses for each of those groups. And then in the black, it kind of describes how the messenger RNA is made and how their genome is replicated. But what's important here, I don't expect you to memorize all these different groups, just that they're, the way they are classified is by their genome and the process of making messenger RNA. Because once messenger RNA is made, then you can transcribing and transcription can take place, so protein is made for the virus. What is interesting here is a couple of these groups do bring along an enzyme of their own, and this is group 6. It is a single-stranded RNA, and HIV is an example of this group. They bring along reverse transcriptase, and that enzyme will basically take that RNA that's their genome and then transcribe it into a DNA. And then the DNA, using enzymes of the host cell, will be transcribed to messenger RNA. It's kind of a backward way of making messenger RNA, or making messenger RNA. But these viruses aren't strategists. Well, actually they are. They figure any way, given the genome they're given, any way they can infect a host cell and use their genome and the host cell's stuff in order to make more of themselves. So, they really are. They're really pretty smart, even though they're not living, and they've really learned how to adapt, okay? The second method is basically approaching viruses maybe not as being alive, but as classifying them like they're living organisms. So this is done by the ICTV, or known as the ICTV classification of viruses. And they pretty much, I'll show you on the next slide, they pretty much have kind of followed along the lines of Linnaeus taxonomy in classifying these viruses. And this is the ICTV stands for International Committee on Taxonomy and Viruses. So these are a group of worldwide scientists that their responsibility is to be identifying, classifying these viruses, and new viruses, there are many of them these days lately, into groups. And the groups are taxon, and it's consistent with taxonomy, and basically there's no kingdom or phylum. The highest group or taxon is order. This may have been updated since this slide was made. There may be some other higher classifications now, but we'll go by this slide. There's no kingdom or phylum. The highest taxon is order. So in order to name these viruses, starting with the highest taxon, order, the last part of the word will end with virales. Family or subfamily, the end of the word will end in viridae or virinae. So if you see that at the end of the word, you'll know that that is the family of a virus, okay? Genus and species names are confusing at best because unlike binomial nomenclature, using Latin or Greek derivatives, we basically, the genus and species names of viruses are given letters and numbers, okay, or very common names. And I'll talk about that in a second here. Bacteriophages, they have their own taxonomy rules, so they don't even follow this scheme of naming or classifying. That's something that probably is a whole course in and of itself. Actually, virology is a whole course in and of itself. You're kind of getting it in a week or half a week lecture, okay? So anyhow, genus and species, let me give you an example of this. We do have COVID-19. That is actually the name of the disease, okay? The coronavirus disease. And it was given the name, the disease is given the name by WHO, the World Health Organization. But the virus that causes this disease was established by this committee, and it is SARS-CoV for coronavirus 2. So SARS stands for respiratory syndrome, CO for coronavirus, and then 2. So that is, it's both quote unquote genus and species for the COVID virus, okay? Okay, now I'm getting to the end here, and I'm hoping this doesn't freeze, because then you're just going to get a picture of my frozen face. I figure, spending 2-3 times recording this is starting to get on, it's starting to wear me down. So finally, the vast realm. We all have, like I said, we've got bacteria, fungi, and viruses that make up our microbiome. Anywhere you go, okay? I know some of you, even on your skin plates, when we were streaking those and they started to grow, may have gotten a little yeast, fungi, or there may have been some molds. Molds are slow growing, so now, if you went back and look at your plate and you see something really hairy growing, that's the fungi, okay? Many times on our skin, that's just picked up. It's like a transient. It kind of hooks a ride on our skin. But we do have viruses that are in our gut. And the thing is, they don't know a lot about the viruses in our gut or on us. They're still studying them, but they would like, first of all, to be able to identify these viruses and group them, because in that way, once they're identified, they may know how they work and be able to do further studies on what effect they have on us, on our biome. And the hope is that maybe we'll identify or focus on some therapies that will benefit us. For instance, if we have a bacterial infection, and if we could introduce a bacteriophage that would be able to kill the bacteria, just infect and kill the bacteria, instead of giving chemical therapies, would that be safer and healthier in the long run for us and for our microbiome? So anyhow, this last slide, and I haven't frozen yet, so this is going to give me cause to celebrate. This last slide here is showing you the research that has been done on each of these types of microorganisms pretty much since 2006. And you'll see way at the bottom there, not very much was done on either bacteria, fungi, or viruses and our microbiome. As the years passed, you'll see that by 2011, there were about 164 research articles on the microbiome. And then we're just starting to see a beginning on research on viromes or viruses in our microbiome. Finally, by 2015, this is the latest data I have, you'll see that there are many, many articles on the microbiome, really more or less on the bacteria within us. Some on the fungi, but it does seem like it's substantially growing in research on the viruses within our microbiome. And as time goes on, we're going to find more and more out about all these microbes, how they work within us and on us, how they contribute or how they harm us. And maybe at some point be able, through nutrition, different types of therapies, be able to promote healthy microbiomes so that we can have quality life and quantity of life. So anyhow, I'm going to end there, and I appreciate you watching this. This information will be on your next exam, not on exam 1. I will focus on the viruses for your exam 2. So anyhow, have a good day, stay safe, and I will see a few of you tonight. Bye.